Greetings, friends, fellow explorers, and potential descendants of Martians? What? Welcome to Ask An Astrobiologist, the show that celebrates the science and the scientists involved in our quest to understand the nature of life in the cosmos. I'm your host, Dr. Graham Lau, and we're brought to you by the NASA Astrobiology Program and SagaNet.org. And this month's episode is going to be really awesome. We are chatting with an astrobiologist who has specialized in how we can use remote extreme environments on our planet Earth to better understand how we could find life out there. Before I introduce our guest, though, I'd like to just give a shout out to all of you out there in Twitter land, on social media, sharing us through emails and talking to your family and friends about our show and sharing this wondrous nature of astrobiology with us. This month, I want to give a special shout out to one Twitter user, at NickBE24, who asked a question on our Twitter poll from NASA Astrobio about how can one become an astrobiologist? What was your path after high school? This is a very important question and one that we get very often on this show. And if you have very specific questions about the career of today's guest, please ask those during the Q&A a little bit later. But for Nick and others who are interested, you can go to the NASA Astrobiology website, specifically at astrobiology.nasa.gov/career path suggestions with a hyphen in between those three words. That'll show you our career path suggestions page from NASA Astrobio, which will help you kind of guiding yourself forward and thinking about how to pursue the right courses in high school and college and to think about your future career. Now, all of that said, I'm very excited to introduce today's guest. Dr. Alfonso Davila is joining us. He's a research scientist at NASA's Ames Research Center, an expert in biosignatures and life detection, remote environments, and searching for life elsewhere. Alfonso, hello, and thank you for joining us for Ask an Astrobiologist.
Hey, Graham. It's nice to be here. Thanks for having me.
I'm so glad you could join us. One thing I love with our guests, we like to showcase your work as a researcher, but we also like for our audience to get to know you and some of your own career path. As I mentioned, we have a lot of young people who want to know how to become astrobiologists. You started off in your undergraduate and master's degree studying marine science, marine physics, marine geology, and then you earned your PhD at the University of Munich, where you studied in geophysics, and then transferred into the SETI Institute and then NASA's Ames Research Center. I'm wondering for our audience, though, if you can tell them, what really drove you into this career? What was it that really gave you that first impetus to want to pursue these sciences?
Well, most of all was chance. I'm not the poster child for somebody who wants to do astrobiology. When I went to college, I went to college at the same time as the Allen Hills meteorite paper was coming out in the mid-90s. So astrobiology was not it became mainstream all of a sudden, but it was a mainstream when I was in high school, for example, or in school. And even when I was in college, it didn't make it all the way to Europe, so to speak. It was probably growing in the US, but it was internationally still not that huge field that it became afterwards. So I had it very clear in my head that I wanted to do science for a number of reasons. I chose a science path in high school, and then through college, as you just said. When the opportunity came up to do a PhD in Munich, I thought, sounds like fun. It was on a very interesting topic that had nothing to do with my undergrad, even better. And so I went there, and that's when, towards the end of my PhD, I got in touch with astrobiology. I was approached by people at NASA Ames, Chris McKay and Imre Friedman, who back then was working with Chris at NASA Ames, and they were looking for somebody with a certain line of expertise that coincided with what I was doing in Germany. And they invited me to come to the US for 15 days, do a very kind of a pilot study. And I came and I thought it was the funniest thing I've ever done in my life. We went looking for fossils in the desert, in the Mojave Desert. And then I learned about astrobiology at that point. It was towards the end of my PhD, and I thought it was the coolest thing ever. It was a chance to put into practice all the multidisciplinary science that I had learned in college, geology, physics, biology, chemistry, and whatnot, and then some sprinkles of geophysics that I learned in my PhD to answer a profound question that was very inspiring to me. Is there life on other planets? I didn't know you could get paid to do that. That was a realization at that point. And then, so I came to the US to do a postdoc at NASA. That was the outcome of that pilot study. I did a NASA postdoc for 3 years, and then I continued on. I decided, okay, enough wandering around. This is what I want to be. I can spend 50 years of my life searching for life on other planets. Seems like a worthwhile thing to do. And here I am.
Fantastic. And now you're an expert as an astrobiologist. That's fantastic. I think that's the case for many of us when we first start college and maybe even through graduate school don't quite know that we're on the path to becoming astrobiologists. I just had a young student just this morning reach out to me from India. She wanted to know if I had a list of all the master's degrees programs that have astrobiology in the title. And my response was, most of us who are astrobiologists don't have degrees that have astrobiology in the title. We come from many different disciplines working together. And so it's really cool to hear about your pathway and then going into geophysics for your PhD. But then you've also now become an expert in analog environments. So not only looking at fossils in the Mojave for that pilot study, but you've now traveled the world and you've gone to places like the Atacama and Antarctica to do field research. I wonder for our audience if you can give them an understanding of why you think it's important for us to go to these systems to look for life elsewhere.
Well, this is a great question. What analogs gives you is a way to experience firsthand conditions that resemble conditions on other planets. Uh, experience them as an individual, like with your inputs, not through the eyes or the ears of a robot, through your own, and also to look at them this multidimensional space that is the natural environment, not just whatever data comes from one instrument, but multiple instruments that you're using all the time. So analog research gives you the complexity, natural complexity of things. It also gives you a close-up view of what the life of a microbe on a place like Mars might be like today or in the past, which is very different from the environment you experience as a human, even in an extreme environment. So learning to see the world from the perspective of a microbe, that's very important, and especially important in an extreme environment where conditions around the microbe can be very different from the conditions a meter away or from the microbe. That's another one. And the third one is that it gives you the opportunity of test scientific ideas and hypotheses, and also test technology that one day you might want to send to other planets to search for evidence of life, for example. Test, especially the technology is an important one, particularly some types of technologies like sample acquisition systems that need to operate on very weird terrains that are not normally found in your backyard. That's analog research can get you that nice connection to whatever planet you want to explore.
It's a very good point. And a lot of our research, when we start preparing for sending rovers and landers to other worlds, we test them a lot in our analog environments here on Earth. A lot of folks might remember the famous picture of Carl Sagan standing beside one of the mock-ups of the Viking landers in the desert in the Mojave. And we've done tests with lots of our other instruments, our rovers. I'm part of the University Rover Challenge, where we have undergraduate students every year bring their Mars rovers they've built out to the desert in Utah, and we give them challenges like they're on Mars. And I understand when you were in the Atacama, you also worked with a rover study doing some drilling in the Atacama called ARADS, or Atacama Rover Astrobiology Drilling Studies. I wonder if you could speak to what that mission was about for our audience.
Yeah, ARADS was a very interesting project we did it's the last big project we've done in the Atacama so far, just before COVID. And it was a multi-component project with the science and technology development. The technology part was to equip a rover with a few light detection instruments, as well as sampling acquisition and sample preparation systems to simulate what a sampling and light detection campaign might look like or might be executed on Mars. We were using the Atacama as a place where we could challenge the instruments to search to find evidence of life in soils that have very low biomass, extremely low levels. So we're really pushing the limits of detection of the instruments, and there we're challenging the drilling system and the sample acquisition system to go through unusual geological substrates where you can have very strong changes in hardness, for example, of the substrate as you drill down. So we can test for failures in the drilling system. We can also test for software recovery actions and whatnot. Um, and then we can also test for mobility for the rover, especially when it comes to sending the rover to a place where a scientist sitting 3,000 miles away or 300,000 miles away might think is a good, interesting spot, and see how we can learn to tell the rover exactly where to go, where to drill, how to collect the sample, and then analyze it with the instruments. So it's a very nice opportunity to do end-to-end science and sample technology testing.
That's very important. And one thing you said there that kind of stuck out to me was the issue with drilling through different substrates. As our audience may be aware, we've had some issues drilling on Mars already. We've never been able to drill very deep. And even with InSight, we were really hoping to get down nice and deep. And we've had some issues with drilling. But drilling really gives us access to the subsurface environment, especially with the very oxidizing surface of Mars. If you had to guess, how deep do you think we really need to drill to get a very good fresh subsurface sample on Mars?
Well, an informed guess would be at least a meter deep. And that has to do primarily with the long term constant radiation exposure of the regolith cosmic radiation, which is something, it's radiation that comes out of the sun and also from the universe surrounding us, high energy particles that they don't come in high abundance, but they're constantly pummeling the Martian surface. And like other types of radiation, like UV, which only penetrates a skin layer of the surface, that radiation can penetrate deeper. And as it penetrates, it actually cascades into the subsurface. And we have very good models that can predict how far that radiation might go. And they tell us that for timescales of millions of years, they typically go a meter deep, 2 meters deep. And so if we want to get away from material that has been radiated to those levels, it's relatively fresh. That's especially relevant for microorganisms or biosignatures that might degrade with radiation. Then 1 meter is probably a good starting point. Otherwise, as deep as you can.
Yeah, as deep as you can would be nice. It'd be nice to have a drilling rig, an apparatus for going deep, but 1 meter does sound pretty awesome if we can get that from Mars. And I do want to remind our audience the reason we're talking about the Atacama, this is the driest desert on the planet. I've never been there myself. I've heard really cool things about exploration there, especially as a Mars analog. We've been looking at deserts for a long time, dry places as Mars analogs, because of the history of Mars. Mars had what we think was a very wet, ancient history in the Noachian, but as we move through the Hesperian and Amazonian, we kind of come to this area where Mars today is very dry. I'm wondering if you can give our audience kind of your vision of what Mars looked like maybe 3.5 to 3.9 billion years ago.
I think it looked like the Atacama Desert. As you say, the Atacama is the driest place on Earth. It's still very wet compared to Mars. We sometimes will lose this perspective. We think that the driest place on Earth might be a good analog to present day Mars. It is not. It's about if you run up the numbers, the Atacama is about 100 to 1,000 times wetter than the surface of Mars. And so, in my mind, the Atacama is a good analog or a good window into what Mars might have looked like towards the end of that water-rich period, as you said, towards the end of the Hesperian, when there were still pockets of liquid water on the surface, but the planet was already cooling down and drying up significantly. That's where I think the Atacama sits. As you move farther back in time, then you can you can think of other analogs on Earth that might fit better than the Atacama. You could think of environments in the Arctic, which are still dry, but you have ice-covered lakes. That might be a better analog for places like Gale Crater, for example. And if you go back in time, then you can travel to more benign environments on Earth where you have temperatures closer to above freezing and whatnot. But the Atacama is, I think, that sweet spot of the tail end of Mars being a relatively wet planet and then just going into extreme dryness.
Yeah, absolutely. And now we don't know if there is life on Mars or ever was life on Mars, where we're going to find those samples. There are some great targets, like in Jezero Crater, looking at a potential river delta could be a great place to look. The subsurface or maybe lava tubes could be great places to look as well. There's a lot of places for us to explore. If any of you watching have questions about Dr. Davila's favorite places to look on Mars for life, please do ask right now. You can ask in the chat on Facebook or on SegaNet. Those questions are coming in for me for our Q&A here in just a little bit. But I have a few more questions yet of my own. First, I want to share a poll that we did through NASA Astrobiology on Twitter. We mentioned, given your background in traveling to the Atacama, Antarctica, and these other field sites, we mentioned that you were in Antarctica testing some novel drilling techniques, and we wondered for our audience if they kind of understood what that'd be an analog for. And we gave them the options of Venus, Mars, or Earth's moon. And the primary answer, with only about 100 out of 200 votes, was Mars. But we also had a pretty decent breakdown of votes for Venus and Earth's moon as well. So I'm wondering if you can let our audience know, Alfonso, why you went to Antarctica and why you were drilling in this site in Antarctica.
Right. That's a very good example of where an analog can really help you develop instrumentation for other planets. In this case, we went to Antarctica because of its connection to Mars. As you say, Graham, Mars is a very cold planet today. It's so cold that most of the surface of Mars never goes above freezing. Maybe except for a thin layer of the surface, the ground stays below freezing all the time. We call that type of ground that never warms up above freezing, we call it permafrost. And permafrost is not uncommon on Earth. There is permafrost in the Arctic, there is permafrost in the Antarctic, there is permafrost in high mountains. It's not uncommon. But Martian permafrost is fairly unique in that it's actually starts with a layer of soil that is dry, but it's frozen. We call that dry permafrost. And then in some places of Mars, if you dig deep enough, then you start to find permafrost that contains ice. It's ice-cemented permafrost. And this transition from dry permafrost to ice-cemented permafrost, which is very common on Mars, is extremely rare on Earth. Most permafrost on Earth has the top is wet, and then at some point you find the permafrost layer that is frozen. But the top is either frozen or is a muddy slush. That's what you find in the Arctic. In Antarctica, the dry valleys of Antarctica, especially the ones that are high elevation, which are the coldest. That's one of the few places on Earth where we find the same type of permafrost we see on Mars: dry permafrost and then ice-cemented permafrost. And when you're testing technology, it's very important to get that transition and the strength that goes with it, the material strength that goes with it. It's important that your instruments go through it if you want to really test them in an environment that is very similar to what you will find on Mars. So that's a unique trait of the Antarctic dry valleys.
Oh, very cool. Yeah, and I can definitely testify to being in the Arctic and using a hand-powered drill to drill some geological samples and having the drill bit get stuck in that wet permafrost. As soon as the drill got stuck, within a split second, the drill bit was frozen in place because that wet permafrost trapped it.
Exactly.
Yeah, so definitely different kinds of drilling regimes we have around the planet in permafrost.
And that's precisely one of the things we wanted to test is how do you drill into permafrost that is so cold that it never thaws? And the number one rule you want to avoid, which is number one rule, as you said, is avoid high temperatures. When you drill, you want to keep the ground and the drill bit as cold as possible so that exactly what you just said then doesn't happen, that you thaw the ice and then it flash freezes and then you lose your mission. So that was one of the lessons that we were learning as we were drilling in Antarctica.
Yeah, and it's a very important lesson, and it's why we go to these places and we test our instruments, we test our tactics and techniques and methods, because we don't want to have something happen on Mars where we have the mission fail because we weren't prepared, which is very awesome. I wonder if you can speak now to the Icebreaker Life mission concept, and what the idea was behind that mission concept, and what you think of that kind of going forward for future research.
Icebreaker was precisely what motivated the trip to Antarctica, or the expeditions to Antarctica to test the drilling. Icebreaker is a mission concept. The idea is to go back to Mars, to a place where there is ice-bearing permafrost close to the surface. Think about the Phoenix lander that went to Mars in circa 2008, and just by scraping a few centimeters off the top of the regolith, it discovered ice very close to the surface. Ice is a great thing for microbes because ice can be a source of liquid water if temperatures are high enough. And so Icebreaker would ask the question, is the ice-bearing permafrost on Mars a habitat for life, and is it inhabited? Can it sustain life? Is actually life embedded in the permafrost? The general idea about Icebreaker is that while Mars is extremely cold today, and that icy permafrost is frozen, it would not be a good place for a microbe to be today. When you go back in time, the climate of Mars changes, just like the climate of the Earth, and it changes in response of changes in orbital dynamics, obliquity, and whatnot. And some of those changes can trigger a significant rise in temperature, high enough that some of that ground ice could melt. And at that point, then you start to deal with permafrost that is very similar to the Arctic permafrost, where microbes can be frozen for a long period of time, but when the ice melts, they can become active, do their thing, and then go back to dormancy when the ice freezes again. And we think that the last time that such a thing might have happened on Mars was about 5 to 10 million years ago. Not that long ago in the life of a microbe. It's long. It's not a lot of time in the life of a microbe, but it's a long time for us. And so that's what Icebreaker is asking. Could 5, 10 million years ago, microorganisms in the icy permafrost on Mars be active? And can we find the remains of those microbes today?
That's very cool. And yeah, there's so much more we could do on Mars and looking for past life. I also want to discuss some of your more recent work, a paper you just published recently, for instance. Before I do, though, a lot of our viewers, they want to know, what is it like to do field research on this planet? How can they be involved? And also, what is it like to live in the Atacama or to live in Antarctica for a prolonged period of time?
Yeah, so the Atacama and the Antarctic fieldwork experience is very different. It's very similar in terms of the extreme nature of the environment, and a lot of the science we do is very connected, despite they're very seemingly different places. But logistically, they're two completely different worlds. In the Atacama, you can go from the driest place on the planet to a 4 stars hotel and a shower in 45 minutes. So the logistics are relatively mild. You also have roads. You can drive with your car. There's a lot of mining activity. There's human presence all over the place, which is kind of bad for the science, but it's kind of good for your own personal survival, and so on and so forth. In Antarctica is a very different place. It's a very different ballgame. You fly to Antarctica to a station, like Murdo, for example, which is crowded with people, about 1,200 people. Then from there, you deploy on a helicopter to a remote area in the dry valleys, for example, and there you camp for 6, 7, 8 weeks with 5 other people that you learn to hate and hate to love at the end of the field expedition. It's a very interesting human experience where you're deprived of a lot of things, mainly showers, things that make you comfortable. You don't get them. It's cold all the time. It's unpleasant most of the time. You eat what you can out of dry food, and then you do your usual business under conditions that most people would not like very much. So it's a very different experience doing fieldwork in both environments, and both of them are, to me, they're fascinating. Going to Antarctica has been a huge personal experience. Going to the Atacama has been as well for different reasons, but scientifically, both of them have been very illuminating.
Yeah, it's wonderful. And I always recommend to younger people who want to get involved in field research, look for others of us who've done field research and ask us, do you have open projects? Are there available, positions to come along? Do you need a graduate student, who wants to go out in the field and learn more about this? There's a lot of ways to get involved in field research. There's also citizen science programs that allow citizens to go out and do some of this field research as well.
And there are grants specifically to fund fieldwork in extreme environments. The Lewis and Clark comes to mind. The first time I went in the field, that was when I was doing my working on my PhD, and I went to a field site as a field assistant. It had nothing to do with my PhD, but it was a great opportunity to do fieldwork. I'd never done fieldwork before. It was a great experience for me. So that's where you want to start. Get a taste for it. If you still like it at the end, then that's when you might consider doing research in field analogs. The one thing I would say about field science is that field science is not different from laboratory science. Doing science in the field is a skill that you develop through experience, just like doing science in the laboratory. In many senses, it's a difficult skill to acquire because it's not something you're familiar with. It's not something they train you for normally in college. But it's also a skill that is difficult to learn because the more you're in the field, the more you learn about the field, the better your science is going to be. So one and only field experience is not going to cut it. If you want to commit to field work, you're committing to many years of visiting the same site to get these knowledge about the site that then allows you to answer the important questions. So be careful what you wish for. If you want to do field science, sharpen up your camping tents because you're going to use them.
Indeed. I'd like to switch topics just a little bit now. You had a very recent paper out this past year in Astrobiology questioning with the search for life on Mars. Is there the potential for biological transfer between Earth and Mars? And so, could we be descendant Martians, or could we find Mars life that's descendant Earth life through the process of panspermia, through biological transfer? Or if we find signs of life on Mars, could we actually find signs of a second genesis, a second origination, or several originations of life on Mars? I'm wondering if you could highlight for our audience some of the takeaways of that research.
Right. Yeah. So this is not a new idea. The idea that life on Mars might have existed because it was transferred from Earth, or vice versa, that life on Earth came from Mars. That's not a new idea. What I tried to do with this paper was to look at the problem a bit more deeply, and also with a time component. So there's been plenty of studies looking at the likelihood that microbes from Earth might survive a ride to Mars on a meteorite. Typically, those studies they assume modern types of organisms that have the benefit of 4 billion years of evolution to figure out how to deal with radiation, dryness, and things like this. But obviously, life on Earth wasn't like that all the time. So I first went in asking the question: at the time where transferring between Earth and Mars was at the peak, life on Earth was not highly evolved yet. So what does that mean in terms of the survival of the transfer, the likelihood of survival? That's number one. And the other one is uh, so early on in the history of life on Earth, we didn't have the diversity of life we have now. Very early on, we only had pretty much one branch of life. It was all unicellular, barely cell-like looking organisms. And so if those forms of life had been transferred at that point, life on Mars would have looked differently than if more evolved terrestrial organisms had been transferred later on. And I wanted to see what would be the implications of an early transfer versus the implications of a late transfer in terms of what we might find. And then all that, I was interested to think about how can we interpret any evidence we find of life on Mars today. How can we interpret it in the context of an early transfer, a late transfer, or an independent genesis? Because the conclusions we might get out of each one of those scenarios would be very different, fundamentally different. Particularly if life on Mars represented a transfer from Earth, if we could figure that out, that would be very cool. It means that life can transfer between planets. But it wouldn't tell us much about how common life is in the universe. It would still be a local phenomenon. But if we found life on Mars and we could evidence of life on Mars and we could establish there was an independent genesis, that means 2 times independently in the same solar system equals a lot of life out there. Very fundamentally different implications of your results. So I wanted this paper to be an opportunity to think about how we interpret any evidence of life we find on Mars, and to be careful with the implications of any finding that we might want to any implications we might reach with those findings.
Yeah, this is still very profound for people to think about. We don't know if panspermia happened. It's an interesting hypothesis, an interesting idea, but we don't have evidence yet that it's happened. But we do have some very good modeling that it could happen. But I love this idea that, if we do find an alternative origin of life within our solar system, that one, it gives us comparative biology. It gives us a lot that we can do right now in figuring out a more universal theory of life. But it also then tells us that life might be common out there.
It is also, Graham, an interesting thing to consider when you think about what are you looking for when you search for evidence of life. Because if you go in with this mindset of, well, I want to know if life is common in the universe, therefore I want to know if there has been a second genesis of life anywhere in the solar system. That was the equation I just mentioned. Two independent genesis equals a lot of life. Now, what do you need to find in order to tell a second genesis from a common ancestor? Not every evidence of life is going to allow you to distinguish that. And in fact, only certain biochemical traits might allow you to tell a second genesis from a close cousin. And so that is an important thing to consider when you design your missions. What am I going to search for? And And so I also wanted to continue that discussion in the within the science community, so that we don't lose track of those high level questions that we want to answer.
Absolutely. Dr. Jim Pass has asked, with the discovery of life on Mars, be it a fossilized form or from the same genesis as Earth life, it will obviously impact humanity in profound, anticipated, and unpredictable ways. Jim wants to know how you think such a discovery will impact astrobiology as a field and in terms of funding and changes in our selection for future missions.
Well, that's a tough one. It's hard to predict how a program might evolve because there's a lot of components, not just the scientific merit, of course. But I would think that if we found evidence of life on another planet, astrobiology would cease to exist as it existed today, and it would become an entirely different field with a program in itself. I think we would want to study that evidence with as much technology as we could. Uh, if we would want to bring samples back as safely as possible. We would want to pursue the same type of thing now that we know that life can be common, either because it spreads easily or because it appears easily on different planets. We would want to search for it on other places. So I would think the astrobiology program would be its own exploration program if we can all agree that understanding life beyond Earth deserves such an important investment in technology and funding. So in practical terms, I think that would be the big revolution. There will be obviously other outcomes, but that's the main one, I think.
That's really awesome. Yeah. We mentioned earlier you kind of first got into this realm of studying of yourself right around the time the Allen Hills meteorite paper came out in 1996, which that paper kind of created the use of the word astrobiology for NASA. And now we have a NASA astrobiology program. So it is cool thinking forward how the program itself could evolve into its own thing in the future.
We would move from something you just mentioned, Graham, which I think is critical. We would move from life detection to comparative biology. What a great opportunity. The big hurdle is life detection. Once we find evidence of life on another planet, studying it is relatively straightforward. We do it all the time on Earth. We know how to study life forms. Finding them on another planet is the big hurdle. After that, comparative biology will become the new game in town. And I think it would be a fascinating, we will be in for a fascinating roller coaster for many years.
Absolutely. Well, one thing that myself and others think about in our pursuit of trying to find life out there and find some comparative biology to study is whether or not humans ourselves will go out there. Our next question comes from Potato Mind at Mind Potato, great handle on Twitter. And at Mind Potato wants to know what you see the future for humans on Mars. Do you think they'll be living in bubble-like structures above the surface, or maybe underground structures? What do you envision for the future of humans on Mars if we go there?
Well, I always like this idea of going back to the caves for humans, and going to Mars gives you these romantic idea of we came out of the caves, we went to Mars, we went back into the cave, at least for a while because we need shelter and protection from radiation, for example, extreme temperatures and whatnot. But it's a nice symbolism that I always enjoyed. Eventually, you would want to see it's more than a vision of the future. I think it in my mind is a wish of what I would hope humanity would strive to go to as a human as a species. Being confined to your own planet, it's like being confined to your own room. Really, the moment you know that there is other places, restaurants and pubs and live concerts and all these type of stuff going out in the streets, that's a good excuse to go out of your room and explore the world and grow as a person. I think growing as a species, it's just the same. Staying confined to your own cozy room, although it might become not very cozy in the not so distant future, uh, but it's an opportunity to grow as a species and to ask a whole set of new questions that I think we need to ask ourselves when we think about the evolution of the human species. How do we interact with the environment? How do we change the environment in a way that is sustainable, but at the same time is beneficial to us and all forms of life? Even more fascinating is if we find an environment with life in it, with extraterrestrial life in it. How do we deal with the ethical and moral questions related to that? All those questions, I think they're going to help us. Asking those questions is going to help us grow as a species. Uh, and so, just going to other planets is, as humans, settling other planets, so to speak. Um, it's only going to allow us to ask those questions and more profound ones.
Absolutely. And one of our next questions comes from a longtime viewer as well, Tom Caruso, who's watching on Facebook. Tom is often interested in the icy worlds of our solar system and the ocean dynamics, dynamics of ices. And his question for you, you may wish to speak to it, is if you can talk about the dynamic environment of ice brine channels here on Earth and their potential for relating to life under the ice on icy moons or exoplanets.
Yeah, the I'm not all that familiar with the ice brine channels. I know what they are and know where they exist and whatnot. Uh, like every other extreme environment, they're fascinating in that they tell you, they teach you a number of things. One is how creative biology can be at resolving environmental puzzles, especially in extreme environments. The other one is what types of environments we might want to explore to search for evidence of life. When I think about ocean worlds, though, I don't think about brine channels. When I think about ocean worlds, I think of dark environments where sunlight never gets there. Microbes have to fight for every possible source of energy they can find. It's a struggle to stay alive. That's a very different kind of extreme than brine channels, where you have to deal with other problems, but one of them is not energy or food, however you want to slice it. Um, Deep subsurface biospheres, dark uh, environments. That's what I think of when I think of ocean worlds.
Awesome. Our next question comes from Navanil Sarangi on SaganNet. Navanil wants to know what would you look for in a core extracted from the dry valleys?
Well, a number of things. One question we went in when we went to the dry valleys was, remember I told you we had this layering of dry permafrost and then ice-cemented permafrost. So one hypothesis we went in when we went there was that we wouldn't find a lot of biology in the dry permafrost. There's no water for them. And we wouldn't find a lot of biology deep into the permafrost because it's very frozen. But we might find some biology, a sweet spot at the interface between the dry permafrost and the ice-bearing permafrost. We call that the ice table. So one of the things we paid a lot of attention was to study the microbiology in those three regions: the dry permafrost, the ice table, and underneath. It turns out that in the dry valleys, the ice table is not even a good place for microbes. It's just too extreme for them. But then we learned as well that as you go deep into the ice, you're going back in time, because permafrost is also it's depending on how it forms, it can record climate conditions at the time of its formation, including the biology and whatnot. So we could still find biosignatures of life frozen in the ground. We could see chemical biosignatures, isotopic biosignatures, whatnot. And then we also learned about how the ice formed. Did it come from melting water? Did it come from freezing vapor? And we could see differences in them. So you can learn a lot about the environment, the context, and the microbiology at the time of formation.
Well, let's relate that then to Mars. So, user Matteo Bori on Facebook wants to know if we have plans to dig for life or to look for life at the Martian polar caps. And maybe, is that terrain too hostile for rovers and landers? Is there a reason we haven't done that yet?
Well, it's certainly technologically complicated. It's very cold. That's one. But the main problem of going to the polar regions of Mars is darkness. You only have a certain amount of daylight in the year, and then you go into pitch black, extremely cold conditions. So you cannot run on things like solar panels for long periods of time. The Phoenix lander was limited to a few months on the surface precisely because of that reason. Now you can get around that by bringing your own isotopic energy source. But then you have a whole different set of problems because those types of energy sources generate a lot of heat. Now you're mixing heat with ice. Not a good idea. We already discussed what happens when you do both. And so technologically, certainly very challenging. And then on top of that is the terrain. It's not an easy terrain to navigate frozen soil or pure ice. And so that's probably one of the main reasons why we haven't done it yet.
Mhm. Well, like we mentioned earlier, I'd love like a really deep sample drilled from Mars, or I'm really excited by lava tubes. I think, going back to the caves is a great place for human exploration as well as robotic exploration. There's a NASA-funded project called NASA Braille that's been going into lava tube caves using Boston Dynamics spot robots to look around these caves and map them. There's so many cool things in the near future for exploring for life on Mars, on Venus, on Europa, on Enceladus, and all these other worlds of our solar system and beyond. Our next question comes from user at Lou the Rover, great name on Twitter. And Lou wants to know that if money or current technological abilities didn't matter, where would you want to send a probe to check for signs of life right now?
Well, my number one place would be the subsurface ocean of Enceladus, hands down. I don't think it needs a lot of explanation, but if you want a few points there, we know we have an ocean, a global ocean of liquid water. We know there is hydrothermal activity, or there is very strong evidence of hydrothermal activity at the bottom of the ocean. That's important for a number of things. Hydrothermal activity generates the kinds of nutrients that life needs to grow and to exist. And hydrothermal environments are also one of the environments that theoretically could have been the birthplace of life on Earth. So it would allow us to test these very specific fundamental hypotheses whether life can exist in those environments. We know from Cassini that there is organic matter in the ocean. We know there is complex organic matter, and there's nutrients and there's energy sources. So I know personally, I'm familiar with one type of microbe at least who will be a happy camper in the Enceladus ocean. So I don't think we can say the same thing about any other place in the solar system right now. Maybe in the future with other missions like Clipper, we learned a couple of things about Europa, but Enceladus is right now is as good as it gets. It's a wet dream for an astrobiologist, literally and figuratively.
Yeah, getting back to Enceladus would be very cool. For our audience, if you're interested in seeing a really cool tech idea for a potential upcoming mission, go to enfold.org and watch one of our recent webinars delivered by Kaylin Carpenter and Morgan Capel of NASA JPL. Morgan has been on our show before. Morgan and Kaylin talk about the potential for eels, as they call them, these long snake-like robots to climb down the vents of Enceladus. We have some very cool things at NASA Astrobiology in the future coming up here for us. I do want to get to some more questions, though. We actually have a bunch more in my queue here. We have one from @organism19874 on Twitter. And this user wants to know, they say, is the omnipresence of life in extreme environments on Earth dependent on the presence of life in more suitable environments? And this is actually a conversation I've had before with others like Carol Cleland and philosophers of science, trying to figure out, are we kind of fooling ourselves maybe by going to analog environments and finding so much life there? Is it more likely that life started in more clement environments on Earth, and that's why we find it in analog systems? Or do you think it's really, really important to have these analog systems?
Yeah, that's a tricky question. Quick answer is depends on the extreme, and it's not a quick way out, but I'll try to explain myself. Not all extremes are the same, and particularly extreme dryness stands out. Extreme cold also, but extreme dryness stands out from all the other extremes. Here's why: there are extreme temperatures, and there are microorganisms that thrive under those conditions and would die in any other environment. There are extremes of pH, and also we find organisms that thrive in those extremes and would die in more normal conditions. Same with salinity. Microorganisms thrive in very salty environments. There are no organisms that thrive in extreme dryness or extreme cold. Those are very different extremes. The microorganisms that we find in extremely dry or extremely cold environments, those are survivors. They're not thrivers, they're survivors. And that is because, and in that case, those organisms exist in those environments because they exist in more clement conditions. They're very good at surviving. They're not very good at thriving. So to answer the question, yes and no. We need life in normal environments so that some of those organisms can survive in some of the extreme environments, cold and dry. Maybe we don't need life in normal environments to survive other extremes like extreme heat or extreme pH. That depends. That goes at the core of what you just said, what the origin of life or the early evolution of life was like. If the early evolution of life was in extreme hot, then that would tell us extreme hot is not that big of a problem. But extreme cold and extreme dry is a very different beast that often doesn't get recognized.
That's very important to think about, indeed. I do want to switch topics just a little bit to something more in the realm of planetary protection now. We've discussed this potential for biological transfer versus having another origin of life on Mars. And one thing a lot of people wonder, if we find microbes on Mars or say with sample return right now, with Perseverance, we're going to be caching samples bringing those back to Earth to study and analyze, potentially for signs of life. If there is Martian bacteria, is it going to be harmful for us? And so user Sangeet Diman on SeganNet wants to know, especially for crewed missions to Mars, do you think it'd be likely or possible for microbial life there to be pathogenic and potentially dangerous for humans? And should we have a protocol to deal with that?
Well, if it was a common ancestor, if we shared the common ancestor with them, they were close cousins, so to speak, of us. The chances of them being pathogenic to us would be higher because their biochemistry would be a lot more compatible with ours. And so one would want to be careful with that. If they were a second independent genesis, it's hard to tell. We don't know what they will look like, but my guess is that the chances of being a pathogen would be less. Um, so that's another reason why it's important to consider that issue of independent genesis and common ancestry. I would say, though, that before we send humans to Mars, there is very likely going to be a biological hazard assessment of whether we find anything there that can be pathogenic, and there are ways we can do that. And Mars sample return already has mechanisms in place to sterilize the sample so that precisely what this viewer was asking doesn't happen, that there are no microbes floating around in the sample chamber that could be harmful to us. So it's a very important question to ask. It has implications both scientific and programmatic and technological.
Very important. And I think our next question is one that's very intriguing as well, that others have wondered, is should we seed life elsewhere? So user at Small Robot Army on Twitter wants to know, so say we find no life on Mars, which I'm not sure how soon that would happen, but say we find no life on Mars. Do you think it'd be scientifically interesting, and I'll even add ethically responsible, to take Earth life there and specifically target seeding life on Mars?
Yeah, this is a good question. My answer to that question is I've bought Chris McKay's and rationale for this, which is published in a number of papers, which I found to be very logical and very fascinating. And the rationale is that an environment with life has more intrinsic value than an environment without life. Now you can buy that argument or not, and there's a lot more information in some of those papers and other papers, but I did buy that argument. And so I do think that as a species that has the capability of populating other environments with life, it would not only be okay, but I think it would be our obligation to try to spread life as much as we can. Again, with the important caveat that the place is not already taken first. If it is, then that's a whole different game. But as much as I like bare deserts, and places without life, I still like to see a tree every now and then. So yeah.
Absolutely. It reminds me of doing the University Rover Challenge and being out in the desert of Utah, which is very Mars-like, but we still see some shrubs and bushes. And I once watched Disney filming John Carter when they were out there filming, and it was kind of interesting. I watched the movie, and I know that like right beside where the actors were in this Martian desert is plants growing, which is pretty funny.
And you cannot ignore that biology is probably uh, the most phenomenal force in the universe. We often talk about quantum physics and the atoms and things like this, but what you get when you form a molecule that can self replicate and evolve, there's nothing else in the universe that can achieve that the amazing level of complexity. So the more we can crank that machine, I think the more interesting the universe is going to be. 99% hydrogen and helium is not that interesting. And maybe you can sprinkle some DNA into it or whatever, make it more interesting.
Yeah, it's a very cool viewpoint. Life finds a way, right? Our next question, we're going to go back now to some more Mars research. Our next question from Arunava Padar at Arunava2512 on Twitter wants to know what are the future challenges for things like the ExoMars rover and Perseverance, specifically, in your own viewpoint, having done research in the Atacama and the Antarctica.
Well, the challenges are for Perseverance. Navigation is going to be one, and selecting the right samples. The Because one thing you learn in going to the field is that you really need to have this global context before you pick the right samples. On Earth, we don't worry too much about it on Earth because we can always go back and get the sample we didn't get or the one that we missed. That's not what that's not the case in sample return. In Mars sample return, that's a one-way ticket. Well, it's a two-way ticket, but you don't get to do it a third time. At least not in our lifetime. So the challenge there is going to be to really walk that thin line between collecting the samples now and getting enough context so that we collect the right samples. For ExoMars, I think is drilling deep. That's going to be the number one thing. The ExoMars, if I'm not mistaken, the goal is to reach at least 2 meters. A lot of things can go wrong in 2 meters. So that's going to be the main challenge. I'm sure they have the technology to do it, but it's going to be scary at, at the very least.
Yeah, I mean, drilling is hard, and sending a rover or a lander to do drilling is difficult. Even for humans on Earth, it's hard for us to drill with a bunch of people with power and a lot of horsepower pushing these drills. And so it's a very interesting thing to think about. And maybe following on from that, we have another question from user at Astro Augusto on Twitter. Augusto Carbadito wants to know what are some outstanding problems in general in terrestrial analog and biosignature research?
Well, I won't speak for all of them, but I will tell you a couple of things about life in extremely dry environments. Number one, we still don't really understand what is it that limits life in extremely dry environments. You would say, duh, is the lack of water. Yes, but we still find microorganisms in those very dry environments, and they don't have adaptations that are really that um, make them all that much better at using water. At least not different from their counterparts in wetter environments. They're very similar. So we still don't really understand what is it that limits life in extremely dry environments. I have a personal hypothesis, which is that life in extremely dry environments is actually energy limited, which is a bit of a counterintuitive hypothesis because in deserts, if anything you get is sunlight, and sunlight is an extremely powerful energy source for life. But but what's happening in deserts is that you cannot use all of that sunlight. You can only use it when there is water around, and that's a scarce resource. And so a lot of the energy you get in the desert, it goes unused because you cannot use it because you're dormant or you're dry in a dry state. And over long periods of time, I think that can lead to energy starvation. That's an hypothesis that comes out from many years of studying life in extreme deserts, me and other people, and that we're trying to test with some specific hypotheses these days. The other one is what happens to dead organisms in extreme deserts. How do they decay? One bad thing about extreme dryness or extremely dry environments is that not a lot of things can grow, but it leaves behind beautifully preserved bodies. And so there is this um, trade that you can play. Well, I'm not getting a lot of signal, but the signal is very well preserved, versus another environment where you might get a lot of signal, but most of it gets degraded before it can be fossilized. So those trades are also interesting questions. And then the third one is what is between cold and dry? I think cold and dry, lack of water and cold temperatures are very similar in many different ways. And I like to explore how organisms cope with one and the other and whether the coping mechanisms are related. That just give you three of them.
Awesome. Yeah, there's still so much more research for those who are watching who are maybe high school students or undergraduate college students or even graduate students who want to be involved. There's still a lot more work to do in terrestrial analog research and in biosignatures. There's a lot of us doing that work, and we can always use more people, more perspectives, more opinions, and more work, honestly, to continue that field forward. I do want to change gears now. We have a few minutes left, and so I just want to ask a couple of more questions that are kind of just like more fun, open-ended questions here. One, Navanil Sarangi has also asked a question on Twitter. Navanil has asked if you know of an ideal analog system for the Dragonfly mission, which we're sending to Titan.
No, that is way too extreme. The cold, something that has the cold temperatures, the organic content. Uh, I cannot think of one. I mean, in the past, people have talked about what do you call them, hydrocarbon lakes, asphalt lakes, as possible analogs, but it's still a huge leap between that, and those are still water-rich environments. So I don't think we have a good analog on Earth for Dragonfly. Dragonfly is flying blind in that sense, not with a good analog.
What we need to do is take a giant warehouse and just build a giant titanium atmosphere and cold environment inside of it to have a test bed. Maybe Navanil can make some money, maybe a couple $1 billion, and we'll build that. um, I think we have time for one more question. So let's ask this one from T Vinit Reddy at T Vinit Reddy 2 on Twitter. This user wants to know, what would be the next step if we find a habitable exoplanet which is not yet inhabited?
An exoplanet that is, and we can confirm that there is no life in it. Well, I don't know. I would move on to the next one and see if there's life in the next one. A habitable, but not inhabited exoplanet that's sitting millions of miles away. There's not a lot of things you can do with it. You can think about studying an environment that is habitable, why it's not inhabited, but it turns out we might have a lot of those in our own solar system. We might have plenty of environments that are habitable and inhabited, and we can study why. So we could answer that question in our own neighborhood. Personally, with thousands of habitable exoplanets and some of them potentially inhabited, I would move down the list to the next one.
Awesome. Well, Alfonso, thank you so much for joining us for Ask an Astrobiologist. It's been a real pleasure having you here for the show.
Thank you, Graham. It's been a lot of fun. Thank you for having me.
Awesome. And for our viewers watching, we discussed this issue with Alfonso's recent paper, which we'll link in the YouTube video. Could there have been an alternative origin of life on Mars, or are we potential descendants of Martian life, or vice versa? We'd love to hear your opinion on this. Do you think that there could be multiple origins of life in different worlds in our solar system, or do you think we all shared a single origin with biological transfer happening? Let us know on Twitter at NASA Astrobio or at Sagan.org. And then, for all of you who want to stay in the loop with our show, with more things going on in the astrobiology realm, you can always sign up for the NASA Astrobiology Program's newsletter. And also, we now have a new sign-up address for joining the SaganNet newsletter. You can join that at signup.sagannet.org. It's been great having Alfonso on the show. A pleasure, as always, to speak to our entire audience and share the wonders of astrobiology with you. Thanks for joining us. And until next time, remember to stay curious.